Good morning, children. Today we are going to discuss about structure. Okay, structure of which one? Immunoglobulin. Immunoglobulin. Okay, children. So observe very carefully. So there is must and should. We should listen very carefully. Okay. Like this, the immunoglobulin structure will be present. Okay. Now this is called very important. This is called the short chain. Okay. This is called short chain. Okay. Now here connection is there. Here also connection will be there. So here also connections will be there. Okay, like this. What are this structure? This is this particular region. This particular region is called, what do you call that particular region? Antigen. Antigen binding site. Antigen binding site. Okay, this region here, one particular region will be there. This region is called variable. It will be change. Okay, variable region. Variable region will be there. This region, that is called, the chain is called light chain. It is called light chain. Here one bond is there. Here it is disulfide bonds. Disulfide bonds. Like that, two bonds are there. It is heavy chain. Heavy chain. The next one, this is constant region. What is called constant region. It's like this. There will be two parts. The structure will be there. This is which one? This particular area. Okay. This is called, in between the light heavy chains and light chains, connection is there by the help of which one? Disulfide bonds. What do you call this? Are disulfide bonds. Okay. This is heavy chain, light chain, variable region, and what you call here variable region. This is small chain or light chain, disulfide bonds. Here also disulfide bonds, heavy chain, constant region, constant, will not change. Like this, the immunoglobulin structure will be there. So in the 1950s, already we discussed about this, but once again we are going to discuss. In the 1950s, who are the scientists? Porter and Edelman. Porter and Edelman revealed the basic structure of immunoglobulin. So they proposed the immunoglobulin structure. Is which shaped the immunoglobulin? Y-shaped structure. Okay. Next, 4 polypeptide chains. How many polypeptide chains? 4 polypeptide chains are there in the structure. Okay. Next, another important one. 2 light chains. 2 light chains. 2 light chains. 2 light chains means it's a L. Next, what is the light chains weight? What is the weight of 2 light chains? 25,000 daltons. 25,000 daltons. Approximately 214 amino acids. Amino acids. Okay, this is the most important case. Amino acids. Okay, and 2 identical heavy chains. 2 identical which one? Heavy chains. Two identical heavy chains also there. What is the weight? Molecular weight 15,000 daltons. Okay, 15,000 daltons. Approximately what 450 amino acids. Amino acids. See now, here NH2 group is there in the sense here carboxylic group. Here carboxylic group. Here NH2 there in the sense here what will be there? Carboxylic group will be present. Means amino groups. Okay, like that the structure also will be there. The polypeptide chains. Next one. The polypeptide chains. The polypeptide chains are okay linked together by the polypeptide chains are linked together by very important what you called disulfide bonds. What you called disulfide bonds will be there. What are the disulfide bonds? Is it? Yes, bonds. Disulfide bonds will be there. Next, one light chain is attached to okay attached to one light chain is attached to each heavy chain okay. Next, and two heavy chains one light chains are attached to each heavy chain. The two heavy chains are attached to two heavy chains are attached to okay each other to form what you call a Y-shaped structure. Okay, so hence an antibody is represented. Antibody is represented by which one? H2L2. What is that one? H2L2. Next one, very important. The heavy chains have what is that? The heavy chains have okay. The heavy chains have a flexible. The heavy chains have a flexible hinge region. Called hinge region at their approximate middles. At their what is called? At their approximate middles. Okay, children, approximate middles. They have what is called flexible hinge regions will be present. Next important thing is each chain have two terminals. What are telling? I'm telling each chain having okay each chain. What are those? L and H? No, L and H chains. Okay, each chain has two terminals. Each chain has two terminals. They are C terminal. C terminal is also called carboxyl. Terminal and amino are N terminal. Amino are N terminal. Each chain has two regions. Each chain has what you call two regions. Okay, each chain have two regions. So L and H2 chain, both chain have two regions. What are those? Okay, one is variable region. Variable region is also called variable region V. But their C regions are okay. Second one is C region. Okay, region is constant region. Constant region. Constant region. Variable region. Constant region. Variable region and constant region. Okay, children, this is a very important at the other end. At the other end will be there. Okay, next important. Antibodies responding to antibodies. Okay, up to here you should listen. Then next point we will discuss next video. Just you see, observe very carefully. The structure of immunoglobulin. This is heavy chain, right? This particular chain is heavy chain. This is light chain. This is COH, carboxylic group. This is amino group, amino group, carboxylic group. Disulfide bonds, variable region. This region will be changed. This is a heavy chain, right? This is constant region, constant. Here this particular areas are constant, will not change. But here this region is there or not? This region will be changed according to the antigen. So that's why that is called variable region. So here carboxylic group and amino group. Carboxylic amino acid, carboxylic acid, amino group. Protein structure. So who given this structure, this information? That is important. In the 1950s, Porter and Elderman were the scientists. Porter and Elderman given the structure of basic structure of immunoglobulin or antibody. The antibody or immunoglobulin is which shaped? Y shaped. Which shaped? Y shaped. Y shaped. There are in the immunoglobulin structure, how many chains are there? 4 polypeptide chains are there. How many chains? 4 polypeptide chains are there. What are those? 2 light chains. 2 light chains. What is the 2 light chains? Very small. And why their molecular weight? What do you call? Their molecular weight is 25,000 daltons. What do you call? 25,000 daltons. There will be 214 amino acids. There will be how many? 214 amino acids will be there. Next, 2 identical light chains over. No? Next one want heavy chains. Heavy chains are there. 50,000 daltons. Their molecular weight. The molecular weight, okay, molecular weight of heavy chains is 50,000 daltons. There are 450 amino acids in the heavy chains. In the light chain, 214 amino acids. Important bit. The polypeptide chains are linked together by, I said no, disulfide bonds. In between the polypeptide chains, 2 sulfide bonds are there. That is called polypeptide chains. One light chain is attached to each heavy chain. One light chain attached to each heavy chain. 2 heavy chains are attached to each other to form what is called a Y-shaped structure. What is called Y-shaped structure. Antibody is represented by H2L2. Antibody is represented by what is called H2L2. What is H? 2 heavy chains, 2 light chains. 2 heavy chains, 2 light chains. That's why antibody is represented by 2 heavy chains and 2 light chains. The heavy chains have a flexible hinge region. at their approximate middles. Hinge region, middle region have hinge region. Each chain L and H also have two terminals. They are carboxylic terminal and amino terminal. Middle region is hinge region, and the terminals, carboxylic group and amino terminal I said or not, terminals are there. Each chain has two regions, variable changing region and a constant region. Variable region is also there, and also what region will be there? Constant region also will be there. Okay, children, there are in each arm. Okay, now we are going to discuss about some more points about the antibody. Okay, we are discussing about some more points. What are the some more points will be discussed? What are those? Observe very carefully. Okay. Next point. What are those? In each arm of the monomer antibody, in each arm of monomer antibody, okay, in each arm of monomer antibody, the V regions are there or not? V regions, okay, the V regions of the heavy and light, heavy and light chains combine to form chains combine to form very important, this one you should have to remember, form an antigen binding site. What is called antigen binding site. Antigen binding site they will form. Okay, children, this is important. Shaped to fit. Shaped to fit. Shaped to fit. What to fit? This is important. A specific antigen determinant. A specific antigenic determinant. Okay, specific antigen determinant. Next one. Consequently, very important, each antibody monomer. Consequently, each antibody monomer. Okay. Consequently, each antibody monomer has two such. Okay. Two such. Okay. Antigen binding regions. Okay, regions. The C regions. What about the C region? The C region. Okay, the C region means constant region. The C region that forms. This is important. The C region that forms the stem of what you called the stem of the antibody. Okay, monomer, stem of the antibody monomer. Okay, determine the what determines which one? Antibody class. Determine the antibody class and serve common functions. Common functions in all antibodies. In all antibodies. Common functions in all antibodies. The functions of the functions of immunoglobulin immunoglobulin the functions of immunoglobulin are agglutination 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 precipitation precipitation agglutination precipitation opsonization 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 neutralization neutralization. Okay children, very very important. This kind of structures will be there, we will be discuss. Okay children, the structure of immunoglobulin in each arm of monomer antibody. So this is the monomer antibody. Each arm have, which one I said, variable region. Variable region. This variable region of the heavy and light chains combine to form, okay, variable region of light chain, variable region of heavy chain. Variable region of light chain, variable region of heavy chain. Variable region of light chain, variable region of heavy chain. Combined form which one? Antigen binding site. Shaped to fit a specific antigenic determinant. Consequently what? Each antibody monomer has consequently, and each antibody monomer has two such antigen binding regions. So here antigen binding region, and what you called here antigen binding site region will be there. The C regions that forms the stem of the antibody monomer determine the antibody class and serve common functions in all antibodies. So constant region which class is that forms the stem of the antibody? See stem, this is a constant region, no? Constant region. This forms the stem of antibody monomer determine the antibody class and serve common functions in all antibodies. Determine the antibody class. Which one? Either IgG, IgM, IgD, IgE, like that. Common functions in all antibodies. It will be served. The functions of immunoglobulin are agglutination. The functions of immunoglobulin are agglutination and also what you call precipitation and opsonization, neutralization. What you call agglutination, precipitation, opsonization, neutralization. Okay children, one by one we are going to discuss about one by one of each one. What is agglutination? What is precipitation? What is opsonization? We will be discussing now. Okay children. Okay children, should listen very carefully about the antigen and antibody reaction. Antigen and antibody interaction. Okay, the reaction between next. The reaction between an antigen and antibody. Antigen and antibody is the basis of okay, basis for humoral okay, humoral immunity, resistance power, or antibody mediated immunity. Antibody mediated immunity. Okay, children, the reaction between, what I am saying, the reaction between, okay, the reaction between antigen and antibody, antigen and antibody occurs, okay, occurs in 3 stages. How many stages? 3 stages. Okay, children. Number one. What are the 3 stages? Number one, first stage. First stage. What will happen in the first stage? In the first stage, what will happen? The reaction involves the formation of first stage. What will happen? Formation of, what do you call it? Antigen and antibody. Antigen and antibody complex will be formed. The second stage, what you called, the next stage is to visible events. Visible events will be takes place. The visible events like precipitation, okay, precipitation, agglutination, and third one is what you call precipitation, agglutination will be takes place. Next, the third stage is the final stage. What is the final stage? The final stage is include destruction of antigen. Destruction of which one? Antigen or its neutralization. Destruction or its neutralization. Okay, children, observe very carefully. We are discussing about antigen antibody interactions. Antigen antibody interactions. The reaction between an antigen and antibody is the basis. Okay, what you call the antibody mediated immunity is called humoral. What you call humoral immunity, are also called antibody mediated immunity. Humoral antibody immunity. The reaction between antigen and antibody occurs in 3 stages. First stage, for formation of antigen and antibody. See, this is the antigen. So, antibody, formation of antigen antibody complex. Then, which one we can see under the microscope, precipitation, like that, or agglutination will be takes place. Precipitation means all antigen antibodies complex we can see. Agglutination means clump like that it will be formed. Third one, the destruction of antigen. The third one, destruction of antigen and its neutralization will be takes place. First, antigen antibody complex, then agglutination. Then what will happen? Destruction or neutralization of which one? Antigen will be takes place in this reaction. Tomorrow we will discuss some more information about antigen antibody reactions. Okay, children, tomorrow we will be discuss tomorrow. Okay, thank you, children.